online Zoom students, can you hear me? Good. So this, I think, works okay, right? It gives us a break. You get to go move around a little bit. Anyway, questions about that were unresolved from your breakout? Well, then remember that, you know how to find me. Email or my office or both. So let's move on to the material in the next class. Before I do that, I have an announcement. One, and that is that the mid-semester reports are, I think, being made available very soon. And, you know, I don't have an exam data point for you. So pretty much you're all going to get sort of, you know, performing well or some generic statement in your mid-semester report. If you want to know details, go to the Moodle grades, and there you'll see your numbers. Okay. I just want to point out that I am compliant with the system of putting up mid-semester grades, but they're not going to be very informative. The informative thing is on Moodle. Okay, so we're going to transition to talking. Well, we're going to talk a little bit about oxygen, and then we're going to talk about how blood carries carbon dioxide and oxygen. That's where we're going now. There is an equation for oxygen, but you don't have to know it. So our alveolar oxygen is equal to inspired oxygen. This is inspired oxygen minus the alveolar CO2 divided by this thing called the respiratory quotient. And this is a little strange because this number here, the respiratory quotient, depends on what you eat. If you eat a pure carbohydrate diet, a pure carbohydrate diet consumes exactly as much oxygen as it produces in carbon dioxide. 1 mole per mole. And so the respiratory quotient, if you eat pure carbohydrates, is 1. So the alveolar oxygen partial pressure is the inspired partial pressure minus the partial pressure that's being taken up by CO2. Because you're producing as much CO2 as you're consuming in oxygen. But if you have a mixed diet, which most of us do, it's somewhere less than 1. And that's why this equation is not used clinically, because they don't know exactly what you're eating. And these numbers can be pretty varied. Generally, physicians assume, if they do use this equation, they assume that your respiratory quotient is about 0.8. Mixed diet of carbohydrates, fat, and protein. So I will not ask you to work with this equation. In the past, I have asked you to work with this equation in lab, but I think that will certainly be after the next exam, if it happens at all. Let's just put this equation on hold until lab. Let's move on to some other key material. Oxygen transport. Key for oxygen transport by blood. Remember that the movement of oxygen from alveolar gas to blood is just diffusion. But the movement of oxygen by the blood is carried by the fact that hemoglobin binds oxygen. So this is a space filling model of hemoglobin. It's a tetramer of 4 highly related subunits. Each of those subunits has a heme group. There's the name hemoglobin. So this is the globin peptide. This is hemoglobin. And here's the heme group. I'm not going to ask you to draw the heme group. The point is that here, this nitrogen connects this heme to the protein. So this purple thing is just a highlight. It's not a bond or anything. So the purple is not a bond. This nitrogen, well, I'm not. Anyway, there's an amino group that connects the heme to the protein. And in the middle of the heme is an iron ion. This is why iron is a necessary nutrient, is because your hemoglobin requires iron in order to bind oxygen. Four heme groups per hemoglobin, one heme group per globin monomer. This is important. So let's get oriented. This is partial pressure of oxygen. This is the oxygen content in mils of oxygen per volume of blood. This is the percent of hemoglobin that is saturated with oxygen. So here we are at 100 millimeters of mercury of oxygen. So this is alveolar, this is oxygenated blood, right? 100, it's 105 in the alveoli, it's 100 in well-oxygenated arterial blood. And you can see that if we depended on just the oxygen that was dissolved in plasma, we'd have a lot of trouble getting the oxygen we need to our body. But we have evolved hemoglobin, and hemoglobin has a very steep and cooperative binding of oxygen. So at high concentrations of oxygen, high partial pressure of oxygen, our hemoglobin is 98% saturated. You may have read about this in the news, where people who have COVID-19, who are are really at risk, have a low saturation. That's one of the markers of dangerous COVID-19. It's kind of interesting. People who have COVID can feel pretty normal. They don't feel maybe a little fever, maybe a little cough. They don't feel super sick. But if they measure their blood oxygen, which is a really easy blood oxygenation, which is a really easy measure, they're not up here. They're like here at 80, and they're at real risk of dying real soon because it falls off very steeply. So that's COVID. That's where we are now. But normally, if we were to measure your blood oxygen, it would be up around between 95 and 98. Below 90 is abnormal.
Is that why they use ventilators to ventilate you?
They use ventilators because in an effort, what happens is when people are dying of COVID, they are trying to respire, it's not working, their muscles get exhausted, and then they die. If a ventilator is just artificial ventilation, so they put a person on, they basically put them to sleep in an artificial coma. And then the machine is trying to pump air into and out of their lungs. And that doesn't always work either, because sometimes the lungs are so impaired that the ventilator doesn't work. I mean, it works, but it doesn't ventilate the blood. It doesn't ventilate the alveoli. The alveoli are so filled with fluid from the disease that they don't ventilate.
Can we artificially stimulate the diaphragm and all the muscles right here and stuff?
No, we don't do that. We pump air in and we force air in and force air out. So normally, arterial blood is 98% saturated with blood. Arterial blood is 98% saturated with blood. Venous blood is about 70% saturated with blood, normally. So this is, deoxygenated, and this is oxygenated blood. So this is the range where we're working, normally. We get down here and we're dying. This is the normal range between 100% saturation, well, 98% saturation, and 70% saturation. Between PO2 of 100 millimeters of mercury and about 40 millimeters of mercury. That's how we carry blood, oxygen. Our blood carries oxygen bound to hemoglobin. And then the hemoglobin is unbound in the tissues that are demanding oxygen. So let's talk about the oxygen story. Let's walk through the oxygen transport story. Let's do this together. Inspired oxygen. What is this arrow representing? What's the physiological word for inspired oxygen getting into an alveolus? There's a physiological word that you learned just a few minutes ago, and it begins with a V. Ventilation. We ventilate alveoli. That's what this arrow is. So label your handout. This is ventilation. What is this arrow? Oxygen moving from the gas in the alveolus through the type 1 alveolar cell that lines the alveolus, that's not shown, through the endothelial cell that lines the capillary. Right? These are cells that are evolved, specialized for being thin and allowing this process. What is this process called? Diffusion. Thank you very much. The oxygen is dissolved in fluid, in saline, in extracellular fluid. So this is gas oxygen, and this is dissolved oxygen. This has a partial pressure of 100, 105, and this has a partial pressure of 105, or 100. Because they are in equilibrium. What is this arrow? Oxygen moving from plasma into the cytoplasm of a red blood cell, an erythrocyte. What moves oxygen from plasma to a red blood, into the lumen, the cytoplasm of a red blood cell? Not yet. Diffusion. So now we have dissolved oxygen in the cytoplasm. Now, oxygen and hemoglobin do what? Bind. And we have bound oxygen. So this arrow is binding. And how does this, so this is in the lungs, how does this red blood cell get to the tissues, the muscle or the brain that needs it? Blood flow, right? What's not shown here is that we have blood flow, okay? So now we have blood flow. Blood has flowed from the lungs to the muscles or the brain or somewhere that needs it. Oxygen bound, oxygen dissolved. What is this arrow?
Unbinding.
Unbinding, easy. What is this arrow? Diffusion. What is this arrow? Diffusion. What are these purple things supposed to be illustrating? Mitochondria, because that's where we need oxygen as the final electron acceptor in mitochondrial oxidative production of ATP. How does oxygen get into the cell? Diffusion. And how does it get into the matrix of a mitochondria? Diffusion. Mitochondria do not have transport proteins for oxygen any more than cell membranes do. It's all diffusing. So what's the driving force for diffusion? A gradient of partial pressure. So this is moving from high partial pressure to low partial pressure because it's getting bound by hemoglobin. Here it's moving from high partial pressure to low partial pressure because it's being consumed by metabolically active mitochondria. Now there are a few points. I'm going to use this slide to illustrate a point, and then I'm going to skip the next slide. So here is the oxygen binding curve of hemoglobin at normal body temperature. Here, there is a temperature dependence of binding. Here, the yellow is the binding curve at a high temperature. So let's go into a metabolically active tissue. Let's go from the lungs, say at normal temperature, and as we're going into metabolically active tissue, oxygen is unbinding, unbinding, unbinding. And let's say we're getting into muscle that has been working really hard, active. What happens to muscle, temperature-wise, if it's working really hard? It gets hot. We generate heat. So we're going to move from this curve to this curve. And so you can see we're going to unbind more oxygen as we move into metabolically active hot tissues. And so hemoglobin has evolved a mechanism for letting go of oxygen as it gets into the most active tissues. This doesn't work in the brain because our brain doesn't get hot, but it works in our muscles because our muscles do. So metabolically active tissues that generate heat help the hemoglobin unbind its oxygen. The heat helps the hemoglobin unbind oxygen at a fixed PO2. So that's the key point. The next slide is more detail about that. It's the same concept, except that there are two other factors that help hemoglobin let go of its bound oxygen. One is temperature, that we just did. One is acid, and one is carbon dioxide. If you have a metabolically active muscle, it's working really hard, it's generating heat, it's generating CO2, and it might even be doing some anaerobic respiration, generating acid. All three of these things work together to help hemoglobin let go of its bound oxygen in a highly active muscle cell. So, a highly useful evolved system for letting oxygen unbind from hemoglobin in muscle. Sophie?
So it becomes more alkaline?
What becomes more alkaline?
Like, if the pH is more alkaline, then it helps it unbind?
No, if the pH becomes more acid.
Does it?
If the pH becomes more acid, it helps it unbind.
Okay.
Normal is red, more
So-
acid is yellow.
So a decrease of pH.
A decrease in pH, more acid, helps at any given PO2, helps hemoglobin let go of its oxygen. Any given amount of CO2, well, this effect is too small to see, so let's focus on the temperature effect. High temperature helps hemoglobin let go, going from red to yellow, of a certain additional amount of oxygen, because the tissues need it. So that's how we evolve this. unbinding of oxygen is facilitated by heat, acid, and carbon dioxide. Yeah.
Is that sort of how like
No, I don't know. You ask hard questions. I don't know. I don't think so. I think heat stroke is your brain getting too hot, but I need to look it up. So I think dizziness. You asked me about dizziness. I think dizziness is your brain not responding well to pH. Hyperventilation. Your pH is respiratory alkalosis. So that's the alkalosis causes your neurons to be wrong and triggers vestibular, abnormalities that make you dizzy. I think. I still need to look these up. All right, let's do the same story with carbon dioxide. This is harder. We might even finish at the end of this. No, there's some more things I've asked. Where is CO2 produced? What's the process? What's the Self-is process that produces CO2? Krebs cycle and pyruvate oxidation. What is this arrow illustrating? Diffusion. What is this arrow illustrating? Diffusion. Diffusion through capillary endothelium into the plasma, blood plasma. Okay, I don't know where this one's going. Well, some of the CO2 stays dissolved. So this is dissolved CO2, dissolved gas in plasma. That's what this is supposed to be illustrating. So dissolved CO2 in plasma. Some of it, what is this arrow that gets partially obscured by this leg over here? What is this? What is causing CO2 to move from plasma into a red blood cell? Diffusion. And some of it stays dissolved in the plasma of a red blood cell. That's what this is. So some of it stays dissolved. But now here's the key point. Well, two more key points. What is this process? Some of the CO2 binds to hemoglobin. And that binding of CO2 helps the hemoglobin let go of its oxygen, which is useful in a metabolically active tissue. But here is the key interesting thing. Well, it's all interesting, right? But here is the key thing. Some of this dissolved CO2 interacts with water, enzymatically accelerated by carbonic anhydrase. That's what CA here is, carbonic anhydrase. This is an enzyme that catalyzes this step into carbonic acid. Right? We've seen this before in an equation, but now I'm putting it in sort of into an anatomical perspective. Carbonic anhydrase, what is this arrow? Carbonic anhydrase does what to become bicarbonate and acid?
Dissociation.
Dissociates into bicarbonate ion and proton. And now you have to change your slide. I want you to put a circle right here. There's a circle right here.
Where?
Right here. These two arrows are coupled to each other. They are not independent of each other. They are coupled to each other. What is that coupling? An antiport, an exchange transporter. We have a bicarbonate chloride exchanger. Because bicarbonate, this is a terrible shortcoming of this image. Terrible. It suggests that bicarbonate can diffuse through a membrane. Well, it can't. It's an ion. Chloride can't diffuse. This is not diffusion. This is co-transport, an exchange co-transport. There's a protein involved in this process. That's what you've got to decorate your handout with. And maybe even go to your textbook and fix it on your textbook. Because even if you sell it, the people who buy it will thank you for making a fix. Put a circle there to illustrate that it is a coupled process of chloride bicarbonate exchange. So now let's see how is the CO2 being carried by blood? It's being carried as dissolved CO2 in serum, plasma. It's being carried as dissolved CO2 in red blood cell, because after all, the red blood cell volume is about 40% of your blood volume. So dissolved CO2 both in plasma and red blood cell. Some of it is carried as bound to hemoglobin. Some of it is carried as bicarbonate inside the blood cell. And some of it is being carried as bicarbonate in the plasma. So some of your CO2 becomes bicarbonate. And doesn't just become bicarbonate inside the cell, it becomes bicarbonate in the plasma. That's key. Bicarbonate is a pH, it's a part of the pH system, but it's also part of our ability to carry carbon dioxide. Okay, how do we get from a tissue capillary to a lung capillary? Blood flow. Abby.
So what ion is driving the transport?
It's,
Is it chloride?
so in this case, it is bicarbonate moving down its concentration gradient, driving chloride. In this case, it's the bicarbonate ion that is driving this antipoison. So unlike some of the other antipoisons that we've talked about, like sodium-driven neurotransmitter co-transport, this one is going to go both directions. And in this story, it's the bicarbonate going down its concentration and chloride going up its concentration. Does that help? Blood flow brings the blood to the lung. So let's tell the story. What is this arrow? Going from this to this. Dissociation. Unbinding. What do we need here? We need the antiporter here. We need a circle here to indicate that these two arrows are coupled to each other through the chloride bicarbonate exchanger. Chloride is going down its concentration gradient, and bicarbonate is going down its concentration gradient, and chloride is going, I think, up its concentration gradient, but I'm not even sure. It doesn't even matter, really. Well, we know that it does matter. Bicarbonate is going down its concentration gradient because it's going to get consumed into this carbonic anhydrase reaction into CO2. So let's go back to this. This is the exchange protein. We need the protein here to get bicarbonate from plasma into a red blood cell. It does it through a protein that exchanges bicarbonate and chloride. We have lots of chloride everywhere. What is this arrow? Chemical word. Begins with an A. Nope. This arrow right here. You take the bicarbonate and protons and making carbonic acid. Nope, that's here. What's this? This is just association. Association of bicarbonate and protons. Now, carbonic anhydrase is catalyzing the conversion of carbonic acid into water and carbon dioxide. What is this area? Diffusion. What is this arrow? What is this area?
Ventilation.
Ventilation. So the driving force for all of this was the fact that this CO2 is lower than this, diffusion. This CO2 is lower than this, diffusion, pulling this equilibrium this way, pulling bicarbonate into the cell, pulling it into carbonic anhydrase to become CO2 because the CO2 is being drawn out through diffusion and ventilation. Okay? So that's the story of blood, of carbon dioxide transport by blood. So the key point here is that bicarbonate is a pH buffer and a way to transport carbon dioxide. Something like 70% of the carbon dioxide that we carry from tissues to lungs to get rid of it is carried as bicarbonate. I won't ask you that number. Just, it's not mine, that's the point. Okay, so here's that Davenport diagram. We talked about metabolic, let's just focus on this arrow here, metabolic acidosis. High activity of, you're exercising, some anaerobic exercise generating lactic acid, metabolic acidosis. Respiratory compensation. Your respiratory system responds. You've all experienced this. You are sitting here, you're breathing, you may not even be aware of it. You go out and you start, running a 5-minute mile, if you can, and you can't do it without changing your respiration. You start breathing really fast, you start really breathing really deep. You automatically start ventilating a lot. And that's probably one of the questions on the problem set with me swimming at a pretty strenuous pace. How does your body know that it needs to increase its respiration? It knows it because we have chemosensors. We have chemosensors that detect oxygen, partial pressure of oxygen, and indirectly they detect partial pressure of CO2. So where are these, so chemosensors, this is now getting into the nervous system, but we need to do this now because this is more part of the respiratory system than the nervous system. Luckily, anatomically, the chemosensors are in the same places as the baroreceptors that you learned about in the cardiovascular system. They're in the aortic arch, and they're in the bifurcation of the carotid artery. So some of the cells in the aortic arch are baroreceptors, and some of them are chemoreceptors. The point is we have cells in our arterial system that detect partial pressure of oxygen and partial pressure of CO2 indirectly. I'll explain what that means in a minute. So the peripheral, and we have two general classes of chemoreceptor cells. Some of them are called peripheral because they're located in the bloodstream, or in the cells that line. They're not in the blood. They're in the endothelium of the aortic arch and the carotid bifurcation. Periphery. We also have chemoreceptors that are called central because they're in your central nervous system. They're in your brain. So central chemoreceptors are in your brain, and peripheral chemoreceptors are in your blood, your vasculature. Yeah?
Are chemoreceptors receptors on the surface of chemosensor cells?
Okay, so we have to be clear of whether we're talking about chemoreceptor cells or chemoreceptor molecules. I'm just talking about chemoreceptor cells. These are cells that express a receptor for oxygen and indirectly CO2. So the chemoreceptor cells are located in the endothelium and in the hypothalamus of the brain. the chemoreceptor molecules are actually located intracellularly of those receptor cells, because the ligand, oxygen and CO2, can diffuse in.
And do you use chemosensor interchangeably?
For this purpose, let's use chemosensors and chemoreceptors interchangeably. So the hypothalamus, those of you who took AMP1 should immediately go to here for the hypothalamus. This is where the central chemosensors are. I will not do neuroanatomy to you, for you, with you in this class. That's AMP1. But that's the hypothalamus. So let's talk about, here's a flow chart. Let's talk about this flow chart from your book, because it walks us through this control. So let's say we have, so this is an increase in alveolar PCO2. Are we hypoventilating or hyperventilating? Are we ventilating too little or too much if alveolar CO2 is higher than normal? Hypo. More CO2 in the alveoli means we need to be ventilating more. So this is hypoventilation, because we need to ventilate more. Hypoventilation, just in this example. Increase in arterial PCO2 because of diffusion. High CO2, let's do the peripheral story first. High PCO2 by, CO2 interacting with water, becoming carbonic acid, generates arterial acid. The peripheral chemoreceptors, chemosensors, detect acid. So these are acid detecting cells. They detect acid, increase the frequency of action potentials, which goes to the medulla of the brain. So there are neurons. There are neurons that go from the peripheral chemosensors, send action potentials to the medulla. The medulla has nerves that control respiration. This arrow, I want you to decorate the slide. This arrow is the phrenic nerve. That's the most important nerve from the medulla to the diaphragm that controls respiration. Yeah, phrenic. Let's see, I want to make sure people are, so. That's the phrenic nerve. Did you see that online? P-H-R-E-N-I-C. Controls the diaphragm. So, the medulla, increasing action potentials in the phrenic nerve, increasing the strength and the frequency of contractions of the diaphragm, increasing increasing ventilation, which returns, remember, increasing ventilation is going to lower PCO2, which will correct for this particular case, right? This is returning CO2 towards normal. That's what it says now. Okay, let's do this. It's the same story with the central chemosensors. Extracellular blood pH, extracellular fluid pH goes up because for the same reason. Extracellular fluid is in contact with blood after all. Central chemosensors detect that pH and increase the action potential frequency, tell the medulla, so in this case, it's the hypothalamus telling the medulla to work to increase the frequency. More contraction, deeper contraction, faster contractions, more ventilation, returning CO2 to normal. So your body detects, so for example, if you are exercising and not increasing your ventilation, your CO2 goes up, your acid goes up, your body detects it and says, we need more ventilation. So that's natural. The point is you have peripheral chemosensors that detect it, central chemosensors that reflect it. The medulla integrates that information. The phrenic nerve tells the diaphragm and other cells to work harder to increase ventilation. So that's the story of control. This is just a piece of that slide with a clicker over here. So I'll let you work on this independently. I'll just read it to you first and then let you work on it. Chemoreceptors, chemosensors, have a channel that detects pH. It's called the acid sensing ion channel. Okay, so there's a channel that detects acid. It's not a channel for protons. It's a channel that detects protons. Remember, detecting protons is what increases the action potentials in these chemoreceptor cells. So I'm asking you, what would a channel that detects acid and increases the action potential frequency, what ion would that be selected for? Let's just go ahead and do it. We got some time. Which one? Sodium. Sodium, increasing the permeability of sodium would depolarize, bring the neuron closer to the threshold, and increase the action potential frequency. So yeah, so sodium is the correct answer. All right. So now, we have done this story. No, we have done this story. CO2, acid, CO2, acid, increasing the firing of central and peripheral chemoreceptors. There's other ways to make acid, which do the same thing. So this arrow is kind of boring. This arrow is kind of interesting. The peripheral chemoreceptors, in addition to having acid sensing channels, have an oxygen sensor. The central chemoreceptors don't. So if on exam 2, I ask you a simple question, what's a key difference between central and peripheral chemoreceptors? Here's the answer. I've told you that I will not ask you that question on exam 1, but I'm not promising not to ask that question on exam 2. And now we have a record of it. The camera has a record of it for all time. But again, oxygen, low oxygen, increases the firing rate of these neurons, increasing the medulla, diaphragm, contractions, ventilation. One last slide, and then we're done. Here is a cartoon of the medulla. I'm not going to ask you about this, so you don't have to take notes. The point here is that there are parts of the medulla, those of you who took AMP1, you can see these are cranial nerves. And here's part of the medulla. So part of the medulla is, remember, this is getting information from the central and peripheral chemosensors, causing you to increase ventilation. So here's the increased ventilation of the phrenic. But you know that you also flare your nose when you are trying to breathe hard. You may not even be aware of it because it's medulla. It's automatic. You start to mouth breathe, You do that automatically. You don't even think about breathing through your mouth, but it happens. Because it's all part of this medulla driving you to open your mouth, cranial nerve 5, open your nose, cranial nerve 7, phrenic nerve, other nerves that control other muscles of inspiration. So, questions?
So for the, if we want to decrease ventilation, would the sodium ion channels just stop working, sensing sodium ions, and then we have the reverse of the entire thing?
I'm sorry, say again?
So if we want to decrease ventilation.
If you want to increase ventilation?
Decrease ventilation.
If you want to decrease ventilation, so you would just tell the reverse of this story. So low CO2, low acid, that is to say high pH, would turn down the firing frequency of these neurons.
Okay, so for the channels, are they integrated sodium channels?
No, this is so the acid sensing ion channel is not a voltage gated sodium channel, but it is sodium permeable.
Okay, so do they inactivate or?
No, they are ligand gated, they do not inactivate. Other questions? Okay, remember you have a problem set due, and the sooner you turn it in, the sooner I'll get it back to you. So I will post the answers to it on Saturday, no matter what.